conus medullaris through this area here, and then all these little rootlets are going to be part of the cauda equina, and then the pia mater comes off the tip of the conus medullaris and continues down and anchors into the bone, and so that's your inferior connection inferiorly to anchor the spinal cord, so it stays in position. Any questions on that? So as we look at protection of the spinal cord, it's very similar to what we see in terms of protection of the brain. We're going to have the bone, which is going to be the vertebrae that surround. We're going to have the meninges again, so we're going to have our dura mater, our arachnoid mater, and our pia mater, same as before. And we're also going to have cerebral spinal fluid that's going to protect it as well from jolts. So as we look at the different meninges, we're going to start first superficially, and we're going to have the dura mater. Now the dura mater in the vertebral canal surrounding the spinal cord, as I mentioned yesterday, is different than what it is when it's inside the skull. Remember, when it's inside the skull, it has the two layers. It has the parosteal layer and the meningeal layer. When that comes down to the foramen magnum, the parosteal layer does not extend through there, but the meningeal layer does. That meningeal layer comes down into the vertebral canal. So that means that this meningeal layer is not up against the bone. It's not up against the bone. It's only the meningeal layer. It's not up against the bone. And remember that this dura mater is going to continue down past where the spinal cord ends. All three of them do that and are going to terminate in the area of S2 and become a ligament that attaches into the bone that then helps to anchor everything. Because, and we'll mention this again in a minute, here is your dura mater. It's not up against the bone. That means your epidural space now is an actual space. And we'll talk about what fills that in a minute. Deep to the dura mater, and actually right up against it, is going to be the arachnoid mater. Very similar to what we saw before. Again, it goes all the way down to the S2 level, just like the other three too. And then attached to the actual spinal cord and giving it this very shiny appearance, impossible to dissect it off, is going to be your pia mater. As we know, the pia mater is going to extend past the conus medullaris as the filum terminale and come down and anchor the spinal cord inferiorly. But we also want to anchor the spinal cord laterally as well. We don't want it kind of wobbling back and forth. So there are little extensions of the pia mater that come over and attach laterally. And these are called the denticulate ligaments because they look like little teeth coming over. And it's just pia mater that goes off laterally and helps to anchor the spinal cord laterally so it can't move too far to the right or to the left. So this pia mater ends up anchoring the spinal cord to different locations. It does so laterally through the denticulate ligaments, and it does so inferiorly through the filum terminale. So in this dissection, we now know that because we've gone through the bone, this is going to be the body of the vertebrae that lets you know this is the anterior. Spinous process that lets you know this is going to be posterior. Here we see the spinal cord, and on the surface of the spinal cord is going to be our pia mater. We then have our subarachnoid space, and then here is going to be our arachnoid mater, and then right outside of that is going to be our dura mater, which is not up against the bone. There's an actual space that's the epidural space now. And so again, as I mentioned, all three meningeal layers go past where the spinal cord ends. So here's our conus medullaris. We're at the level about L1, L2. Although the spinal cord is in, our dura mater, arachnoid mater, and pia mater are going to extend down. And we know that the extension of the pia mater is called the filum terminale. Well, all of that connective tissue is going to get together at the level of S2, and what it's going to do is going to form what's called the coccygeal ligament, which is going to connect to the coccyx, and so it's going to anchor that spinal cord inferiorly. Well, if we have our three meninges, let's talk about the three spaces that are associated with it. So we know that most superficially we have our epidural space. Deep to the epidural space is going to be the dura mater. Deep to the dura mater is going to be the subdural space. Deep to the subdural space will be the arachnoid mater. And deep to the arachnoid mater is the subarachnoid space, and then our pia mater. So what about these spaces? the three meningeal spaces are still here. Whether they're going to be an actual space or a potential space, it depends. So we have the epidural space. It was a potential space in the skull because you had your dura mater directly up against the bone. But now with the dura mater away from the bone, the epidural is an actual space. And that actual space is filled with epidural fat and a venous plexus. And so we're going to see that this epidural space is between the vertebrae and the spinal cord's dural sheath, this dura mater. It's an actual space. It's filled with that epidural fat and the vertebral venous plexus. And this venous plexus is going to be bringing blood superiorly, and it's actually a way that cancer can spread superiorly towards the brain. The fat, on the other hand, is very useful as that fat is easy to visualize in MRIs, and so it's helping to actually visualize the spinal cord through different imaging processes. Now, the epidural space around the brain is not continuous with the epidural space around the spinal cord. Okay. So what happens is that dura mater is affixed to the foramen magnum. Now that's important because if you get a hemorrhage or a hematoma in the epidural space, if it's around the brain, it cannot go past the foramen magnum to go around the vertebral canal, or vice versa. So that epidural space around the brain, which is a potential space, is not continuous with the epidural space around the spinal cord. If you get a hemorrhage or hematoma, it can't exchange between those two areas. And that's the same with the subdural space. The subdural space was a potential space around the brain, and it remains a potential space. This remains a potential space. So that's because deep to, and remember the subdural space is that space between the dura mater and the arachnoid mater. Remember, deep to the arachnoid mater is the subarachnoid space, which has CSF, and that CSF is going to push the arachnoid mater right up against the dura mater. So it is a potential space between the dura mater and the arachnoid mater. Now you can get a hemorrhage or hematoma that can open that up, but normally those two are right up against each other. We also then have the subarachnoid space. The subarachnoid space is between the arachnoid mater and the pia mater. It was an actual space around the brain. It remains an actual space around the spinal cord, and that's where the CSF is located. And that subarachnoid space around the brain is continuous with the subarachnoid space around the spinal cord. So if you get a hemorrhage or a hematoma in one area, it can spread to the other, and vice versa. Okay? So related to this, we have two punctures or blocks that you, I'm sure, have heard of. We have a lumbar puncture, spinal tap, or spinal block, which is going to be associated with the subarachnoid space. And we have an epidural block, which is associated with the epidural space. So let's talk about each one of these. In a lumbar spinal puncture, so in a lumbar puncture, spinal tap, or spinal block, a patient's asked to lean forward and hug their knees. Now they can be laying on their side or they can be sitting up, but they're going to hug their knees. And what that does is it opens up the space between the vertebrae. So it's going to, the flexion of the back leads to the laminae and the spinous processes of the vertebrae coming apart, allowing for the insertion of a needle between the two different vertebrae. The area is anesthetized and cleaned. So this is going to be posterior, or sorry, this is going to be this is going to be anterior, this is going to be posterior. Here's the needles here. The skin is going to be anesthetized and cleaned in that particular area. And with that lumbar puncture, the needle is going to go in along the midline between the spinous processes of L3 or L4, or between L4 and L5. Now, why do you think you want to go in at that level? Why would you want to go in here?
Because it's not at the spinal cord.
Spinal cord ended up at L1, L2, so you don't have to worry about hitting the spinal cord down at that particular level. And so that needle is going to go through it's going to go through the skin, it's going to go through the the fat tissue underlying, it's going to go through any muscle tissue, it's going to go through the dura mater, it's going to go through the arachnoid mater, and then it's going to go into that subarachnoid space. And that little subarachnoid space at that level, remember, is called the lumbar cistern because there's no spinal cord at that level. The only thing you have going through there is the filum terminale in the cauda equina. Well, you don't have to worry about hitting those because if you ever try to make spaghetti and you try to get that one noodle out with a fork so you can test that spaghetti noodle, every time you put that fork in, the noodles move to the side. It's sort of the same idea here. So a lot of those nerve roots, filum terminale, are moving off to the side as that needle approaches into that area. Now that you have the needle into the subarachnoid space, into that lumbar cistern, you now can measure cerebrospinal fluid pressure, so you can get the pressure of that area. You can withdraw CSF for biological and chemical examinations, which are important in evaluating a number of central nervous system and meningeal disorders. You can look for the presence of red blood cells. You should not have red blood cells in your cerebrospinal fluid. Remember, CSF is made from blood plasma. Red blood cells should not be in it. You can also put an anesthetic agent in here, and that's called a spinal block. And put an anesthetic agent in here, spinal blocks take effect in about 1 minute, and then anesthetize everything inferior to the waist. And that anesthetic agent is going to remain in this area as long as the patient is somewhat upright, because that anesthetic agent is going to be heavier than the cerebrospinal fluid and stays in that particular area. If the patient tends to lay down, that anesthetic agent can head towards the brain area and actually cause a headache, a spinal headache. Well, the other type of block is an epidural block, and as the name's telling you, it's a little bit different. Yeah?
So is this space continuous with the brain?
It is. So it's continuous with the subarachnoid space around the brain, and that's why they end up getting a headache if they try to lay down after they get a spinal block.
Okay.
Yeah. So the other type of block is an epidural block. The name's telling you things are a little bit different here. An epidural block, the name is telling you that the needle is only going into the epidural space. And so if you're only going into the epidural space, that means you're not going as far, and you shouldn't have to worry about hitting the spinal cord. So everything is done the same as before, but when you put the needle in, you just don't go as far. You're going to end up putting the needle into the epidural space and putting the anesthetic agent into that fat tissue that's in that area. And that anesthetic agent impacts spinal nerve roots in the vicinity of the injections. But epidural blocks take about 10 to 20 minutes to take effect. So about 10 to 20 minutes to take effect. Here you can see a little bit more of a close-up. This is dealing more with the spinal block. And so this is going to go all the way into the subarachnoid space, and so you can see here how you're going through the skin, you're going through the subcutaneous tissue, you're going through ligaments, muscle, and so forth between the vertebrae. You're going to go through the dura mater, the arachnoid mater, and go into that lumbar cistern. And now that you have the needle in there, you can check pressure, you can put something in, you can take things out, and do a lot of different tests. And here you can see that that distance is actually quite large. If you ever get one of these, don't look at the needle. So I gotta tell you, I had a student, I was giving a lecture, you know in the lab sometimes we keep water and Kool-Aid up at the front in case anybody gets a little bit lightheaded. I'm giving this lecture and I just see this hand come up and over the podium and grabs the Kool-Aid and the water and pulls it down, and I'm just like, okay. Afterwards he came up, he's like, I'm so sorry, I was remembering, I had a flashback to my wife had this done to her when she was in labor, and he almost passed out, so he was not enjoying, he remembered all of this and the length of the needle, and he was just done. Okay. So this next part actually starts to look at the actual spinal cord, and we're going to take a look at it, and we're going to actually draw some of this out on Monday, but let's talk a little bit through these images first, and then we'll do our arts and crafts on Monday. So when we look at the spinal cord, there's that basic pattern that I mentioned, and this is coming from the posterior portion of the neural tube, and we keep the lumen of the neural tube, and that's going to be your central canal of the spinal cord. That's surrounded by gray matter, and that gray matter is in this H-shaped or butterfly-shaped pattern. H-shaped or butterfly-shaped pattern. Remember, the gray matter is composed of things like neuronal dendrites, some unmyelinated axons, lots of neuroglia, and a lot of neuronal cell bodies. Now, the white matter is found superficially. And when we look at the white matter, we're gonna see that that white matter is gonna be organized into what are called tracts or fasciculi. And those tracts or fasciculi are gonna go two different directions. So here's a close-up. This is looking at the central canal of the spinal cord. You can see that they're lined by this population of cells. What's this population of cells?
Ependymal cells.
Ependymal cells. Those are those ependymal cells. It contains cerebrospinal fluid, and there's the cilia associated with those ependymal cells to help circulate the cerebrospinal fluid. And remember that central canal of the spinal cord is continuous with the fourth ventricle. So CSF can come down from the fourth ventricle into the central canal of the spinal cord, but it's got to get back up to that central canal of the spinal cord, get up to the fourth ventricle again, because remember the fourth ventricle is the only location that you can get from the internal areas out into that subarachnoid space. And so we have that median aperture and those two lateral apertures to move through at the fourth ventricle. Now, as I mentioned, Leslie's gonna go through these tracts or fasciculi with you on Monday afternoon in lab, and that's that other little lecture that is posted on Carmen. It says continuation. And what I'm gonna tell you is just that this white matter is organized into tracts or fasciculi. And so when we look at this is incredibly organized. And on this side of the image, they're showing you the motor tracts. On this side, they're showing you the sensory tracts. Sensory tracts, which again are on both sides, sensory tracts are bringing information up towards the brain. So they're gonna be bringing information up. So those are gonna be ascending tracts. The motor tracts, on the other hand, are coming down from the brain, and so those are gonna be descending tracts. And a lot of times these tracts are named based on where they're coming from and where they're going, which makes it very nice. Like the corticospinal tract. We've seen this before. We saw it as the cerebral peduncles in the midbrain. We're gonna see it as the pyramids in the medulla. And here it is coming down, but it's already crisscross sides, so this is actually controlling skeletal muscle on the same side. But corticospinal tract, cortico for cortex, is referring to the primary motor cortex. In spinal, the information's coming down to the spinal cord. So the name is actually telling you where the information begins and where the action potential is gonna end. If you look at things like the spinothalamic tract, this is spinothalamic, so spinal cord, thalamus. Remember that most of your sensory information, almost all of it, is kind of through the thalamus before it comes up to the cortical areas. And so this is going from the spinal cord up to the thalamus. This is telling you it's an ascending tract automatically. And again, Leslie will extend that information when she meets with you next week. And when we look at, what I'm gonna concentrate on more is the gray matter. And when we look at that gray matter is very organized. And so when we look at that gray matter, we can see that we have that gray matter in this butterfly-shaped pattern, and we have these wings of the butterfly, which are called horns. We have posterior horns, and we have anterior horns. Now for this particular unit, we're going to ignore the yellow for right now. The yellow is gonna come into play when we talk in the next unit about the autonomic nervous system. But right now, we're gonna be looking at this posterior horn and the anterior horn. The posterior horns are going to have interneurons in them. And those interneurons have sensory information come into them, and those interneurons will then use those ascending tracts to get information up towards the brain. So their axons become the ascending tracts towards the brain carrying sensory information. That sensory information is actually coming in on an unipolar sensory neuron. And so that information is coming in. You can see that there's a little bump on this dorsal root. This is called a dorsal root ganglion. And in there are the cell bodies of those unipolar sensory neurons that are bringing information in from sensory receptors at the surface of the skin. So this is sensory information coming in, comes into the back of the spinal cord, into the posterior horn, and then these interneurons are going to take this information and take it up towards the brain so that you can begin to perceive it. In the front, on the anterior side, are the anterior horns, or also known as the ventral horns. Those anterior horns or ventral horns contain motor neurons. Those motor neurons are going to get information coming into them from the primary motor cortex. And so they're then going to send that information out the ventral root into the spinal nerve and then out to the skeletal muscle they control. So these are gonna gain that information coming down from the primary motor cortex and then send that information out. Now, if we take a histological section of the spinal cord and look at the posterior horn or the anterior horn, what's going to be the shape of these neurons? Does anybody remember what the structural classification of these are? So if you think about interneurons or association neurons, anybody remember what their structural classification is?
Multipolar. Multipolar.
Multipolar, they're multipolar, good. What about in the anterior horn? Those motor neurons, what are they?
Multipolar.
They're also multipolar. So histologically, they look exactly the same, but you know what they're doing based on their location. If they're back here, they're an interneuron or association neuron. They're gonna be conducting information around the central nervous system. If they're up in the front, if they're on the anterior horn, they're going to be sending information out to the skeletal muscle to control it. And remember that we are gonna form 31 pairs of spinal nerves. And so really, to form those spinal nerves, oops, go back. Remember that, so what you have is, coming out is the axon of that motor neuron. Going that way. You have the sensory neuron bringing information in. So the two combine in the intervertebral foramina to form the spinal nerve. So it's a mixed nerve that contains both information. But there's a division of labor here that takes place. Information only flows this way along the dorsal root and out on the ventral root. But once you get to that spinal nerve, it's mixed. So we're forming 31 pairs of spinal nerves, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. And it's those dorsal and ventral roots that are coming together in the intervertebral foramina that forms the spinal nerve. And that spinal nerve is really short, and then it's going to split into the dorsal and ventral rami, which are also mixed. Remember, as these roots come off, whether it's dorsal or ventral roots, they come off first as little rootlets, and we saw that in the pictures earlier. Then they form the actual root, and when you see this in laboratory, you'll see how they come off as little rootlets first and then get together. And here you can see that. So this is a posterior dissection. You can see the little rootlets coming off, and then they're going to come together and form the dorsal root. So these come together to form this dorsal root, these come together to form the dorsal root here, and so forth as you go down. And it's on both sides. Coming off between the dorsal and ventral roots, look what's coming out right here and right here. There's those denticulate ligaments. And those denticulate ligaments are formed from pia mater that's anchoring the spinal cord laterally, so it can't move too much one direction or the other. Okay. So what we're going to do on Monday when we come in is we're going to draw out a cross-section of the spinal cord and look at the neurons and the jobs that they're doing. After that, then we'll start talking about special sensitives. Anybody have any questions?